Hi everyone, I am Dr. K.SHAGIRTHA, Assistant Professor, Department of Biochemistry, St.Joseph College of Arts and Science, Cuddalore. In this video, we are going to talk about primary cell culture. Now, what is primary cell culture? Primary cell culture is a culture. First, cells are taken from organism and put into a artificial media and turned into a clone of cells. Let me select an particular organs like pancreas, liver, kidney or brain cells. Take the cells directly and place the cells into the media and allow the cells to grow. These are the cells termed as primary cells. The whole process is known as primary cell culture. Thus, the primary culture is the initial culture before the first subculture. The term cell line is used for the propagation of culture after the first subculture. Primary culture broadly involves the culturing techniques carried following the isolation of the cells but before the first subculture. With the experience of the personnel working in tissue culture laboratory, the following characters are considered for efficient development of primary culture. First one is embryonic tissues or rather than adult tissues are preferred for primary culture. The quantities of cells used in primary culture should be higher since their survival rate is substantially lower. The tissue should be processed with minimum damage to cells for use in primary culture. Selection of an appropriate medium is advisable. For the addition of serum, fetal pollen sources is preferred rather than calf or horse serum. It is necessary to remove the enzyme used for disaggregations of cells by centrifugations. These are the techniques used for primary cell culture. Among the various techniques devised for the primary culture of isolated tissues, three techniques are most commonly used. They are mechanical disaggregation, enzymatic disaggregation, and primary explant techniques. First, we discussed about the mechanical disaggregation. In this disaggregation of soft tissues, example spleen, brain, embryonic liver, soft tumors, is usually employed. These techniques basically involve carefully chopping or slicing of tissues into pieces and collection of split out cells. The cells can be collected by two ways. First one, pressing the tissue pieces through a series of sieves with a gradual reduction into a mesh size. Second one, forcing the tissue fragment through a syringe and needle. Although mechanical disaggregation involves risk of cell damage, the procedure is less expensive, quick and simple. So next one, enzymatic disaggregation. Enzymatic disaggregation is mostly used when high recovery of cells is required from a tissue. Disaggregation of embryonic tissue is more efficient with higher yield of cells by use of enzymes. This is due to the presence of less fibrous connective tissues and extracellular matrix. So now we discussed about the trypsin disaggregation. The term trypsinization is commonly used for disaggregation of tissues by the enzyme trypsin. The crude trypsin is more effective due to the presence of other protease. Cells can tolerate crude trypsin better. So the crude trypsin is very important. In this trypsinization method, we have two more separations. This is a warm trypsinization and this is a cold trypsinization. Now we discussed about the warm trypsinization. This method is widely used for disaggregations of cells. The chopped tissues is washed with dissection basalt salt solution and then transferred to the flask containing warm trypsinization 37 degrees centigrade. The content are stirred and at the interval of every 30 minutes and supernatant containing the dissociated cells can be collected. And then after the removal of trypsin, the cells are dispersed in a suitable medium and prescribed keeping on ice vial. Cold trypsinization. Next method is a cold trypsinization. This technique is more appropriately referred as the trypsinization with cold pre-exposure. The risk of damage to the cells by prolonged exposure to trypsin at 37 degrees centigrade. After chopping and washing of tissues are kept on vial with soaked with cold trypsin for about 6 to 24 hours. The trypsin is removed and discarded. However, the tissue paste contain residual trypsin. This tissue paste in a medium is incubated at 37 degrees centigrade for 20 to 30 minutes. The cells get dispersed by repeated pipetting. The dissociated cells can be counted appropriately and diluted and then used. The cold trypsinization method usually results in a higher yield of viable cells with an improved survival of cells after 24 hours of incubation. Next one is a collagenase disaggregation. Collagenase disaggregation is one of the enzymatic method. Collagen is the most abundant structural protein in higher animals. It is mainly present in the extracellular matrix of connective tissues and muscles. The enzyme collagenase can be effectively used for disaggregation of several tissues that may be sensitive to trypsin. So we select the tissue and add to the antibiotics, chop the tissue pieces and washed by settling. After that, incubated in a medium containing collagenase for 1 to 5 days. Pipetting the dispersed tissue and separation by settling. And finally, we wash to remove the collagenases. Now we got fibroblastic cells and then epithelial tissues. Collagenases disaggregation has been successfully used for human brain, lungs and several other epithelial tissues, besides various human tumors and other animal tissues. Addition of other enzyme hyaluronidase promote the disaggregation. Collagenase in combination with hyaluronidase is found to be very effective for dissociation of rat and rabbit liver. This can be done by perfusing the whole organ in situ. Use of other enzymes in disaggregation, mainly trypsin and collagenase are the most widely used enzymes for disaggregation. Certain bacterial proteases like pronase have been used with limited success. Besides, hyaluronidase is also used in conjugation with collagenase for effective degradation of cell surface carbohydrates. Next technique is a primary explant techniques. The primary explant technique has undergone several modifications and is still in use. The simplified procedure adopted for primary culture is given below. The tissue in basal salt solution is finely chopped and washed by settling. The basal salt solution is then removed. The tissue pieces are spread even over the growth surface. After the addition of a fresh medium, incubation is carried out 3 to 5 days. Then the medium is changed at weekly interval until the substantial outgrowth of cells is observed. Now, the explant are removed and transferred to the fresh culture vessels. The primary explant techniques is particularly useful for disaggregation of small quantities of tissues, example skin biopsies, The other two techniques, mechanical or enzymatic disaggregation, however, not suitable for small amount of tissues, as there is a risk of losing of cells. So the primary explant technique is very useful for this. Next one, applications. Applications mainly in cancer research, virology, toxicity testing, vaccine production, genetically engineered proteins, replacement tissues or organs, drug screening and development. So first we discussed about cancer research. Difference between normal cells and cancer cells can be studied using primary cell cultures, a screening of overexpressed or underexpressed markers leading to oncogenic activation can be detected using primary cell culture as a model. Moreover, using primary cells as control model for anti-cancer research is significantly exploited. Next one is a virology. Primary cell culture are used to replicate virus and can also be used to detect and isolate virus for studying their growth and development cycle. Primary cells are also used in virology to study the mode and mechanism of virus infection. Toxicity testing. Primary cell culture is used to study the effective new drugs, cosmetics, and chemicals on cells. Primary cells are also used to determine the maximum permissible dosage of the drugs. Next one is a vaccine production. This is very important in primary cell culture. Cultured primary cells are used in replications of virus for production of vaccine. Vaccines for polio, rabies, chickenpox, and hepatitis B are produced using primary cell culture. Genetically engineered protein. Primary cell culture are used to produce genetically engineered protein such as monoclonal antibodies, insulin, hormones, etc. Such recombinant technology is utilized in reshaping many therapeutic. Next one is the replacement of tissue or organ is very important for human beings. Primary cell culture can be used as replacement of tissue or organ in wound healing and other tissue engineering applications. Example, artificial skin can be produced by using primary cells to treat patients with burns and ulcers. Research is ongoing for artificial organ culture such as liver, kidney and pancreas. In this regard, many researchers are talking up stem cell culture as these cells have the potential to self-proliferate and differentiated into several cell lineages. Many therapeutic applications of adult stem cells are currently in trials for tissues, organs, regeneration and replacement technology. The last application is drug screening and development. Primary cell culture are used to study cytotoxicity and safe dosage of new drugs. Primary cell culture therefore play an important role in pharmaceutical industries. Cell culture is a prominent tool in cellular and molecular biology research, often spreading to clinical applications. Cell culture using primary cells can be excellent model system for studying physiology and biochemistry of cells, drug toxicity and metabolism, and other biomedical applications. Primary cell culture is being used by researchers worldwide for their role in maintaining the consistency, efficiency, and reproducibility of result, besides being an excellent mimic model of in vivo physiological conditions. So this application is very important, mainly in focusing on cancer research, virology, toxicity testing, vaccine, different types of vaccine production, and the proteins are generated by genetically method. Replacement of tissue or organ is very important for human being. So we are going to replace our some ulcers. It is very easy. And drug screening and development is also very important to receive the target tissues. Thank you.